A very good afternoon to one and all. Last session of the faculty development program. Now I am proud enough to introduce a speaker, Dr. Murali. Dr. Murali has completed his Bachelor of Pharmacy in the year 2005 from the Tamil Nadu Dr.M.G.R. Educational Medical University, Chennai. He obtained Masters in Pharmaceutical Chemistry from the same university. He worked as a lecturer from 2008 to 2010 in the Department of Pharmaceutical Chemistry at C.L. Bade Mehta College. In 2010, he received a research grant from Professor Piero Silivian of Nanobiotechnology and Bioanalysis Group, University of Rovira, Virgili, Tarragona, Spain. As a research fellow, Dr. Murali conducted a study in the optimization of separation and isolation of circulating cell-free fetal DNA for non-invasive prenatal diagnosis. He had worked with Curie Consortium of Nanotores, and his interdisciplinary doctoral thesis investigated the interaction of nanoparticles with neural stem and tissue type cells. He made a short-time research at NILU, Norwegian Institute for Air Research, Norway, at Bayer Technology Services, Germany, at Institute Catala di Nanocenia Nanotechnology, Spain, at Center for Bio-Nano Interactions, UCD, and at University of Namur, Belgium. In 2015, he obtained doctorate in theoretical medicine from Doctoral School of Semmelweis University, Budapest, Hungary. After his PhD, he spent few months at Magneto-Optical Spectroscopy Laboratory, Budapest, University of Technology and Economics, under Professor Dr. Istvan Kesmarki, working on the magneto-optical diagnosis of malaria. He took his postdoctoral training through Animal Biotechnology Career Enhancement Program at the Translational Research Platform for Veterinary Biologicals, Tanuvas, and then he spent at the Center of Nanotechnology, Indian Institute of Technology, Roorkee, as a postdoctoral fellow. In January 2017, he joined in Laboratory of Pharmaceuticals Nanomaterial Science, Department of Material Science and Engineering at the Technion-Israel Institute of Technology, supported in part at the Technion by a fellowship of the Israel Council for Higher Education for Planning and Budgeting Council Fellowship. He holds several prestigious fellowships, including Marie Curie and National Institute of Health. He is an author of over 30 publications and inventor of patent and patent applications. The underpinning focus of Murali's presentation research is creating personalized solutions that utilize micro and nanoscale technologies to enable a range of theranostic for neurodegenerative disease and brain cancer. He is a pharmacist and he is exploring different disciplines of nanobiotechnology, neural cell biology. In 2020, Dr. Murali received European Union Interact grant to explore his transgalactic approaches on diagnosis and the treatment of pediatric brain tumor. Among numerous other fellowships, Murali has received the Innovation Prize in 2020 from the Startup Portugal for his innovative treatment approaches on COVID-19. At present, he combined his transgalactic ideas to initiate the path in high-reward entrepreneurship. I welcome you, sir, for today's session.
Thank you so much, ma'am. So I hope you all can hear. Thanks so much for a kind introduction. I'd like to thank Hari sir for inviting me to this the virtual seminar. Always, it's my pleasure to give this virtual seminar in the faculty development program, which was organized by Dr.M.G.R. Education and Research Institute. Thanks again. So today my topics, what I like to give a topic, to talk about the nose to brain transport of nanomaterials, a new platform for precision nano-neuromedicine. So previously I thought of giving different topics, but then I realized this is a needy one at present. Corona, like COVID virus, also transporting through nose to brain. Of course, not many patients are affected by neurons. Not many people are affected neurologically. They are not affected by this COVID, but still, this is an entry point. That's why I chosen this topic. So this is a snapshot of a presentation. So so first I'm going to talk about precision medicine, personalized medicine, and individualized medicine. And is it different from usual medicine? My answer is always yes. On where it goes, challenges, and how this should be nano-neuromedicine. And also I'm going to talk about neurological disorders, biological barriers, because our body is full of barriers, and what is a motivation. And then finally, I'm going to talk about non or minimally invasive approaches in drug delivery, which is the important goal of precision nano-neuromedicine. At last, I'm also going to talk about nano safety, like toxicology, risk assessment, which is very important when you're developing any sort of drugs. So nanoscale, hopefully many people you came across about this nanoscale. Our body is is a composite of nanomaterials. For example, proteins is in the nano size, and DNA bases is also in the nano size. Ribosomes, for instance, is a nano size. Genes also in the nano size. And many viruses is in the nano size, around 80 to 120 nanometer size. For instance, there are two different approaches, like bottom-up and top-down approaches for this in the case of nano. Also, many nanomaterials like carbon nanotube, it's 1 nanometers, and the quantum dots also is around 10 nanometers. So a lot of things are nanometer size. For example, a lot of our cells is around 10 micron size. So our body is full of nano. So what is nanomedicine? Nanomedicine is defined as the application of nanotechnology to achieve breakthroughs in healthcare. Also, the nanomaterials, it does enable early detection and to improve diagnosis and treatment and follow-up diseases. So a lot of advantages of nanotechnology. I think many of you came across a lot of advancements in nanotechnology in the disease diagnosis and therapy. So finally, my definition is always it is like nano in medicine is called nanomedicine. So what is personalized medicine? It's in the past 5 years, it's a new mantra of evolving in healthcare. A lot of research center has been established, and a lot of people are started working on this personalized medicine, precision medicine. So what's the difference between, like, how it differs from conventional medicine? So for instance, as you can see here, the conventional medicine is the traditional medicine, one size fits all approach. For example, every patient with the same diagnosis, they receive same kind of treatment, same kind of drug treatment. In case of personalized medicine, the name itself indicating is like a more personalized and more individualized medicine. For example, we give a treatment according to the genetic profile. Like that's what the Human Genome Project, the successful story of Human Genome Project. So each human beings, we are unique, right? So we are height-wise, the weight and color-wise, we are unique actually. So every patient, they receive a different level of therapy. For example, in a traditional approach, everyone gets the same treatment, but in this personalized medicine, we are differentiating into targeted and standard therapy. Because in some standard therapy, some people they will respond, or some people they will not respond to the treatment. So that's what precision nanomedicine is talking about. And what's the major goals of precision medicine? So there are many major goals of precision medicine. The primary goals is like minimally or non-invasive. As you know, the previous approaches like surgery, for example, is a completely invasive procedures. And after surgery, recovery of patients, it takes so many months, days, even years actually. But in case of minimally invasive surgery or minimally invasive drug delivery approaches, it overcomes the conventional approaches. And also cell type specific. For example, precision, that's the main goal of precision medicine. Previously, the off-target accumulations, for example, for example, cancer is the main approach. You can, if you administer the drug, it spreads all over the body, it creates cytotoxicity. But this precision medicine, it's like a main topic, main goals is like cell type specific and low cost and improved outcomes. For example, the India-like countries, we need a low cost treatment system or diagnostic system, because previously most of the diagnostic systems are like really expensive. Not only India, even the developed countries, they are looking for the low cost treatment systems and target specific. For example, the precision oncomedicine. As I mentioned, the number of cancers that's a devastating diseases, but we need a more target specific treatment and also diagnostic approaches. And another goal is treatment without side effects. That's very important. That's the aim of the precision medicine, like with the treatment without side effects. Also, the prevention, the early detection, also very important for the disease diagnosis. Even in coronavirus case, we need to diagnose the disease as early as possible. Then you can treat them more easily. And last but not least, I have to talk about the safety aspects of nano. Not only nano, even the drug aspects like risk assessment, the toxicology. That's very important goal of precision medicine. These are the goal of precision medicine. So now I'm switching to the pharmaceutical R&D. The problems or major challenges in pharmaceutical R&D. As a pharmacist, the biopharmaceutical classification system, like class 1, 2, 3, 4. Among these 4 systems, 2 and 4 is a big problematic, because it's a sad truth. The 50% of approved drugs and 70% of new drugs falls on this particular category. The problem is, I mentioned like our body is full of barriers, right? So blood-brain barriers, skin barriers, intestinal barriers, and many barriers. So in order to cross these barriers, or if you need a treatment on these particular areas, your drug should have a high permeability. The problem is, most of the drugs falls on this category. So where the nanotechnology platform comes to overcome this, how it overcomes the biopharmaceutical problem is, as I mentioned, challenges in drug delivery. For example, the off-target accumulation. As I mentioned before, in case of cancer, if you are administering any drug, it spreads all over the body, it creates a toxicity. So using a nano approach, you can target more target specific, so that you can target one particular cell type or particular tissue. That's the main aim of the nanotechnology or precision medicine. And also drug resistance. This is a big problem. The next big problem, I think the scientific community, we should work on drug resistance. Now how the COVID pandemic started, the same way drug resistance will start very soon. So we need to find a solution for the drug resistance, multi-drug resistance, and antibiotic resistance particularly. And also poor bioavailability. That's another problem in conventional drug therapy. And physical chemical instability. Most of the drugs is not stable inside our body, because his body is full of biomolecules, macromolecules. These macromolecules, in this macromolecules, your drug substances is not stable. So physical chemical stability. So these are the things will be overcome by applying nanotechnology. What are the drug delivery nanocarriers? There are different types of nanocarriers. For example, different based on surface chemistry. You can, positive and negative charge. Also the composition, there are different compositions that are available in liposomes. For example, I think a lot of people are working on liposomes and polymeric nanoparticles, silica, iron oxide, even virus also, non-virulent viruses act as a drug delivery vehicles, and dendrimers, metallic nanoparticles. So these are the different compositions we are using in a drug delivery. And also physical properties, for example, different geometries, I would say. As you can see, different geometries, different sizes, different shapes, it's also used as a drug delivery platforms. Apart from that, different targeting ligand, this is a very popular one these days, is to target specific different proteins, peptides we are using for target particular diseases, and nucleic acids, atoms, we are using siRNA therapy and all we are using in a drug delivery. Also small molecules and polymers we are using in Apart from this, the nanotoxicology is a big question mark. A lot of research is going on in this particular area. And among all other vehicles, our interest for the past 10 years, we are working on polymeric nanoparticles. Why? Because polymeric nanoparticles, this sub-micron ranges 1 to 1,000 nanometers collagen particles. You can able to encapsulate the drug molecules, the pharmaceutical molecules within the polymer matrix. That's one advantage. And apart from that, you can say a number of advantages. For example, is simple production process, easy to produce, and preparation is available to scale up, like large scale mass production is much more possible in polymeric nanoparticles. And also physical stability in biological fluids. That's I mentioned is a drawback, but in using a polymeric nanoparticles, you can able to overcome this drawbacks. Apart from that, you can improve the stability of in enzymatic degradation. For example, the conventional drugs, it's degraded by enzymes, so you can able to improve by coating with polymers. Also, sustained release, that's very important. We need sustained release or control release, and high intracellular uptake compared to the microparticles. You need like a more dosage inside cells. So these are the possible, and also the possible surface modification for targeting agents. For example, you can able to functionalize with different antibodies, ligands, peptides, cationic molecules, so that you can go for the more site-specific target. These are the advantages of polymeric nanoparticles. So far, I discussed about nanotechnology, nanomedicine, precision personalized or individual medicine. Now I'm going to talk about the neurological disorders and what are the biological barriers and what is the motivation of this particular research. As you know, this disorders of nervous system, there are a number of disorders, more than 600 neurological disorders, but very few, they are devastating, and more often people are affected by these very few diseases like vascular disorders, for example, stroke and hemorrhage. This due to high blood pressure and cardiac vascular diseases. Stroke is more often. Even India-like populations, most people are affected by the stroke. In case of structural disorders, you can say groin, spinal cord injury. It happens due to a physical injury, or sometimes happens with the natural genetic issues. And also brain tumors. The brain tumors is not only adult brain tumors, even the kids, the young kids, they are affected by these brain tumors. It's pediatric brain tumor. This is really devastating. And also the functional disorders like migraine, epilepsy, dizziness. Neurodegeneration like Alzheimer's is more popular in scientific community. Parkinsonism, multiple sclerosis, amyloid lateral sclerosis. These are all like neuromuscular disorders, Huntington diseases. So the main problem is, in order to treat all these diseases, your drug molecules or your therapeutic should cross the blood-brain barrier. The brain's own security system is called blood-brain barrier. The problem is, most of the small molecules and almost 100% of large molecules cannot bypass this brain own security system. This is the main problem. That's why scientific communities, particularly neuroscience community and pharmaceutical scientists, are working towards this mystery. And another problem is, neurological disorders cause 17% of global deaths. Apart from that, if patients affected by any neurological disorders, it not only affected the patients, it affected the whole family sometimes. In case of stroke, the whole families will be affected by mentally. So these are the main problem of neurological disorders. And as I mentioned, there's barriers. So first, before I'm going to talk about the blood-brain barrier, there is a very important barrier, it's like solute barriers, which is present in the central nervous system. What is solute barriers? As you know, brain has like an endothelial and also the epithelial barriers. One is like a choroid plexus, which is called epithelial barriers. In the choroid plexus, the cerebrospinal fluid, which is shortly called as CSF. This CSF the present in the CNS, it formed at the rate of 0.5 ml per minute, and its a total volume is 200 ml. The 30 ml which is present in this ventricle region, and remaining it's present in subarachnoid space. Another unique feature of this cerebrospinal fluid is like every 4 hours once it's completely replaced. That's a unique feature of this cerebrospinal fluid. And based on this fluids, we can categorize the blood liquor barrier, which contains choroid plexus, as I mentioned. This contains capillary endothelial and ependymal cells. And next, blood-brain barrier. I think many people we are exploring this blood-brain barrier. For example, blood-brain barrier is composed of endothelial cells, astrocytes, pericytes, and liquor tissue. Liquor tissue is like this, the CSF and tissue sites, ependymal tissue. So these are the barriers which is present in the brain. So that's a big obstacle to develop a drug discovery to treat a CNS disorders. And next, if we're talking about blood-brain barrier, blood-brain barrier is composed of many cell types. For example, it's composed of endothelial cells, which gives a tight junctions, like a complete barriers between blood and brain. And also there's astrocytes and pericytes. These are the cell types present in this blood-brain barrier. Along with it, the microglia, which is macrophages, which is immune cells, the primary immune cell which is present in central nervous system. Also, is a neurons present in a blood-brain barrier. So mostly my talks, I'm going to talk about this microglia and neurons in my later slides in a more detail, like how it is protecting, how microglia is protecting the brain and all. This barrier, it's still is a mystery in a scientific community. It's regulated permeability with many unknown details. For example, glucose is easily crossing the barrier, but same like glucose molecules, it's not crossing. That's a big mystery in a brain anatomy. And particle size, we still don't know. We are exploring different particle size, charge, lipophilicity, chemical nature, different aspects we are exploring. In what aspects brain is responding, and what sort of particles are to be investigated. That's a big question mark still in this neuroscience community. And if I'm talking about the mechanisms of drug transport in the brain across blood-brain barrier, there are different mechanisms behind this. For example, the first mechanism, diffusions, drug molecules diffuse into cells and goes. Another mechanism is paracellular transport. In case of epithelial or endothelial, it's transport between the cells, intracellular space. That is paracellular transport, and transport to proteins. And this is a more popular one. This is like receptor-mediated transcytosis. It's very popular in nanomedicine. A lot of you can surface functionalize specific receptors, specific peptides. You can target particular cells. So this is a very popular one. And adsorptive transcytosis, and during adsorptions from the cells, and it's efflux transporters, it's ABC transporters, ATP binding transporter. That's a big issue for the drug resistance. So this, I think, scientific community should work on this efflux transporters. So these are the main mechanisms of the blood-brain barrier crossing approaches. And next, if I talk about central nervous system drug delivery approaches, there are different approaches. For example, invasive, non-invasive, and advanced approaches. For example, the conventional methods are like invasive, just open intracellular implants. Just open a skull and keeping any implants. That's a completely invasive. So you need a kind of specialist like neurosurgeons, and transient BBB disruption using kind of chemicals like oleic acids, kind of chemicals. You can disrupt the blood-brain barrier. And also intraventricular infusions, as I mentioned, CSF directly, as you can see in the picture, directly they are introducing a drug into CSF. These are all like invasive, and you need a specialist like a neurosurgeons. And more recently, the non-invasive approaches. This is the main target of precision medicine, personalized medicine. A lot of chemical methods, biological methods, particularly the nanocarriers, plays a very important role in non-invasive. And in the non-invasive, the one advanced the approach is like a nose-to-brain transport through olfactory pathway. That's what I'm going to talk in some of my research things. I'm going to talk in this particular pathways. So recently, the nose-to-brain transport is emerged as a new approach to circumvent the blood-brain barrier and efficiently to deliver drugs into the central nervous system. And if I'm talking about the non-invasive, I should emphasize about minimally invasive focused ultrasound-mediated blood-brain barrier opening. I think recently many Indian professors they argued with me that this is not safer, but there are a lot of clinical trials is going on this particular ultrasound-mediated blood-brain barrier openings. A lot of clinical trials you can see, and also a lot of successful stories using this focus ultrasound. And we also initiating some kind of projects with Pittsburgh Medical Center and Vascular Medicine Institute in United States. So this is more focused, as you can see here. You can apply a focal ultrasound, and so that the tight junctions, as I mentioned, the barrier will be opened. The drug molecules should cross the barrier, the brain, to treat any disorder. So this is a kind of minimally invasive approaches, and it's a booming one approaches. And the next the main talk about the non-invasive approaches like nose to brain transport. As a pharmacy person, the advantage of nose to brain because of a lot of systemic circulations is one of the advantages, like non-invasive method, and avoids hepatic first pass metabolism. That's another advantage. You do not need to cross many barriers, and drugs will not be degraded and circumvent the blood-brain barrier, BBB. So it's another very good advantage. And more importantly, this is a painless. Without any single pain, you can administer the drug. And the distance-wise is a very, very smaller, like just nose to brain. And the previous literature precedents, they were talking about the ideal size to cross this neuroepithelium is like 100 to 300 nanometers. So again, I'm emphasizing this point. The international delivery approach is emerged as a new approach to target the brain. And in the year 2004, before that, nobody knows. I mean, the olfactory system is much more important, same like our vision, like, visual system like eyes. In the year 2000, in the year 2004, Professor Richard Axel and Professor Linda Bach, they got a Nobel Prize for unravel the function of one of our senses, the olfactory system. From their investigation, we got an answer for many unknown questions. For example, how this brain connects with the external world, because brain, it's not connecting with any external part. This is the only way, like nose to brain is the only way to connect with the external part. So this is a core philosophy for psychology and neuroscience. And another important is it's how we smell. Same like, vision, the smell is very important, right? So how we smell, these are the questions they answer from the research work. And how does our sensory system work? And how the brain is interact with outside world. So these are the questions has been answered from the research point of view. So that after this 2004, there are a lot of research going on, and particularly in nose to brain drug delivery approaches, and also different approaches. And as I mentioned, nose to brain transport, distance wise, it's a very smaller, like, just nose to brain. But the problem is, the olfactory region tissue is like a tightly packed one. Actually, it's not like just with a single cell. It's like a mix of cells without any extracellular space. As you can see in this presentation, the olfactory bulb is composed of many cells types, like mitral cell and olfactory receptor neurons, and cribriform plate, which is connecting with the choroid plexus. A lot of cells, and also the billions of neurons present in these particular regions. And these neurons are not alone. It always is connecting with the glass cells. It's a kind of delicate system. And this neuron network is dynamically changing environment, and it's a delicate system connections. And neurons, as you know, is like a more fragile in nature, and it's more peculiar option. Another unique feature in olfactory neurons, the lifespan varies from 4 to 8 weeks. So these are the unique feature of this olfactory neurons. And next time I'm talking about nose to brain transport, I think many people are working on in vivo models, right? So when you're working on in vivo models, sometimes you cannot identify many mechanisms actually. So in order to identify the mechanisms. You should work on ex vivo or in vitro aspects. The previous models, nurse to brain transport modeling in vitro is much more important. Actually, there are existing models like nasal epithelium, but those models is not giving much information. So detection of fluorescent particles, fluorochrome analysis, primary cell culture. That's what I'm going to present in my presentations in the later slides. The previous models, the problem is highly simplified and two compartments models, and they didn't use is deprived of many exact cell types which is present in the olfactory bulb. So using the models, we cannot identify exact mechanisms. That's what happens. I like to say this unusual slide I'm presenting now. The previous precedents, like a lot of literature says, the international pathways is to olfactory nerve pathways. But in recently, what we identified, we reported is the nurse to brain transport is not by olfactory sensory neurons. Because those models, they use directly the animal models, but in our case, we use the in vitro models and we analyze the complete mechanistic points reasons. So here I'm presenting my particular research on in vitro aspects like precision personalized medicine aspects. We isolated the olfactory tissue type cells from the olfactory bulb, olfactory epithelial regions, as you can see. And we characterize with different specific antibodies. We isolate different sort of cells here, and we characterize with more biomarkers, as you can see here. And is OV means like olfactory bulb regions, as you can see, the beta-3 tubulin is like a more neural specific markers, and GFP is like a glia specific markers. We characterize thoroughly. We also, this olfactory bulb neurons also specific for it express some kind of growth factors, and also we characterize with the Western blotting, and it's present the olfactory marker protein, which is more specific for the olfactory receptor neurons, and this also tubulin is specific for the receptor neuron. We also characterize the olfactory epithelial cells. There's a positive, is expressing a lot of biomarkers like olfactory marker proteins. Also, we characterize the brain part isolated cells like primary neurons and macroglia and also microglia, which is the immune cells which is present in the CNS. After we characterized, these are the polymeric materials we use. We use 5 different polymeric nanoparticles with a different size between 17 to 500 nanometer size. As I mentioned, 100 to 300 is the ideal size to cross the nose to brain, right? So we use like 17 to 500 nanometer size with the different compositions, particularly like hydrogen polymethyl methacrylate and hydrolyzed galactomannan and PEG, PCL, and Tetronix, which is one of the FDA-approved polymers. So we use it's different surface charged nanomaterials we used. And we also characterized initially for the biocompatibility purpose, and all our nanomaterials are really biocompatible with all olfactory bulb cells, olfactory epithelial cells, and neurons and microglia cells. And after we characterized biocompatibility, we did the nanoparticle internalization studies. So in this case, we used, as I mentioned, we used 5 different nanoparticles in 2 different cell types. One is like olfactory sensory neurons and primary cortical neurons, like brain neurons. As you can see in this picture, the neurons did not take up the particles. Instead of neurons, some other cell types is taken up the nanoparticles. I'm showing only one picture, not showing all other pictures. Just for example, I'm showing here. As you can see, in this primary cultures, the primary cortical neurons and olfactory sensory neurons did not take up the nanoparticles. Instead of that, some other cell types is taken up the nanoparticles. So further, we got the hypothesis that to isolate complete primary brain cultures, which contains 3 different cell types. One is like astroglia. This cell type contain astroglia, microglia, and also neurons. All these 3 types. When we expose the polymeric nanoparticles, contain this green color polymeric nanoparticle, the particular cell types taken up this nanoparticles. So already we proved that neurons did not take up the nanoparticles. So the only two options here is like astroglaya, either astroglaya or microglia. So we also we analyzed the particles are internalized into this cell vesicles. Also, we analyzed, in order to analyze the active cellular uptake, we also analyzed not only in 37 degrees centigrade, because 37 is like physiological temperatures, right? And also we analyzed at the lower temperature, because at this lower temperatures, all the transport system will be suppressed. We also analyzed, as you can see in 4 degree, the particle just only decorated on the surface is not internalized. And further, we identified using a astrocytes. Astrocytes is nothing but it's astroglia is one type of cells which is present in this category. We separated the astroglia, and when we exposed our nanomaterials, polymeric nanoparticles, some selected astroglia, it has like some interactions with selected astroglia. As you can see in the 4 degree, again, it's only on the surface, not internalized. As you can see here, at 37 degrees internalized, but in 4 degree, it's just on the surface. So we confirmed that it's active cellular uptake, and not all astroglia is taken up this nanoparticle. Only some selected astrocytes is taken up this nanoparticles. And afterwards, there is only one choice. We proved that neurons did not take up nanoparticles. We proved that astroglia, only selected astroglia, it's taken up these nanoparticles. And the third option is to isolate microglia from the other cell types. What we did, there's a video how we separated from other cell types. These are when you use certain cell culture media, the microglia stays back, and other cell types it comes off as a layer, as you can see in this picture. And afterwards, this, as I mentioned, is like it's a macrophages. Microglia is macrophages which is present in the CNS. See this macroglia, always it chew up any external materials or maybe any debris which is present in the brain. This is what it's doing in the brain also. It actively is monitoring the brain environment. Any external material goes, this microglia will capture, as you can see. So the unique feature of microglia, after we characterized after we separated the microglia, we exposed nanoparticles onto the microglia. As you can see, both cases, cortical and olfactory microglia phagocytose the nanoparticles, regardless of any functionalizations. So previously, what we found, neurons did not take up the nanoparticle, but instead of neurons, this particular microglia, it taken up this the nanoparticles, regardless of any functionalizations. Also, this microglia, when you expose to any certain nanomaterials, it exposes INS, which is like a nitric oxide synthase, which is a marker for the one is stress cells will express these kind of markers. Also, we did a blotting analysis, and as you can see, the band, of course, it's very clarity-wise is not good, but as you can see very deeply, there's some bands. The particular cell types is expressing this particular biomarkers. Afterwards, what we did, using a transgenic animals, which is more specific for the fructyl ghaalin receptors, GFP-labeled animals, we isolated the microglia, and we exposed the red color labeled nanoparticles onto the microglia. As you can see, microglia catch up this the red labeled nanoparticles. So again, we confirm this microglia is responsible for the uptake. Afterwards, it's one of the serendipitous discovery. Occasionally, I found in my experiment the cell types, when we isolated the olfactory cell types, contain some kind of dots kind of thing, as you can see in the picture. And when we analyzed, the drop is nothing but it's a lipid droplet accumulating microglia. It's shortly called as LDAM. When these cells felt stress, these cells secrete this kind of lipid droplets. It's a kind of new discovery, because previously we know adipose tissue cell types, it's expressing this kind of lipid droplets because that's a unique characteristic, and also some cancer cells it express this kind of lipid droplets act as against mechanisms. But the glia is expressing this kind of lipid droplets against any stress. So when it happens, when they feel like high levels of reactive oxygen species, which is called ROS, and secrete pro-inflammatory cytokines, this particular microglia secretes droplets. The schematic of the droplet contains cholesterol, sterols, phospholipid monolayer, lipid anchors, and extra. And when we analyze ultrastructural analysis, this white ball-like thing, it's a drop, the lipid droplets. So this we proven through ultrastructural analysis. As you can see, the white drop is this lipid droplets. And again, I'm showing this how this microglia is like more active. As you can see, how is capturing any debris is really active in even in the brine environment. This microglia is called as the garbage man. It's on the move and dynamic sensor, because when any external material comes, it comes and catches. And as you can see in the another video, how motile, how active it is, this microglia. So this is a unique characteristic of this microglia. And when we analyze with ultrastructural analysis, as you can see, the microglia and neurons, always the microglia is protecting the neurons, because as I mentioned, neurons is like really peculiar and it's more fragile in nature. It's more sensitive to the external insults. So always, even in the brine environment, this microglia is protecting its neurons. So it has like some specialized areas, it has connections. So same this schematic, again, I'm showing this picture, microglia always protecting the neurons. And we are in our another experiment, what we performed, is one of the lab grown experiment, the mini brines. We exposed to nanoparticles, as you can see, the polymeric nanoparticles on green color. And this is composed of many cell types, which we obtained from the brine regions. And as you can see this video, the particular cell types is taken up this nanoparticle, the green label nanoparticles. The particular particular materials is particular cells is nothing but it's a microglia. We presume that it's a microglia is it acting as a sensor, and whenever the any engineered nanomaterials goes, it catch this particular things, as you can see in the nice video. So this particular places the particles are aggregated. And when we investigated the ultrastructures of this mini brines, what happens when we introduced amphiphilic polymers or ceramic hybrid nanomaterials? It contains polymers and ceramics. And when we introduced this nanomaterials, from the mini brine, the particular this lipid droplets containing microglia, it comes out of this space and capture this the nanomaterials. So in the brine immune system, it can sense, as I mentioned, it's a dynamic sensor. It can sense engineered nanomaterials in a manner similar to the sensing of virus-like pathogens. Actually, why I mentioned this, because even in coronavirus case, it's happening. When virus enters our immune cells, which is present in lung or nose, it activates and it creates a lot of cytokine. That's why it's cytokine storm. It's a big problematic it's a big obstacle for the treatment. So similar way, even in brine, the microglia act as the immune cells. It comes and catch up this nanomaterials. And to sum up our findings, the inherent defense mechanisms in the olfactory region, the mild injury, in case of mild injury, the astrocytes become phagocytes and remove the foreign materials and produce anti-inflammatory cytokines. In case of excess injury, like any harmful materials like virus or any pathogenic materials, if it enters brine, the reactive astrocytes, it got activated and produced pro-inflammatory cytokines. And these cytokines, further it activates the microglia, the primary CNS resident macrophages. and it produces a lot of inflammatory reactions. So our findings reveal that neither olfactory nor forebrain neurons internalize nanoparticles. Conversely, the olfactory and cortical microglia, as I shown before, the phagocytose the nanoparticles independently of the features. So we analyzed with more than 7-8 different nanoparticles, all type of nanoparticles were our phagocytosed by the microglia. And not only in vitro, we also performed in vivo imaging. We performed the non-invasive in vivo imaging. As I mentioned, the precision medicine goals is like, not to harm any animals, like kind of minimally invasive or non-invasive. This is a kind of IV system where you can analyze the biodistribution or particle kinetics without sacrificing the animals. So we also analyze after IV injections, like tail injections, after intranasal, with the different time points. So I'm not showing all the results. I'm showing very few pictures here. And we also found some of nanoparticles, polymeric nanoparticles in brain regions after intranasal. As I mentioned, in the past 10, 11 years, I'm working in the area of nano-neuroscience. So I work with the different neural tissue types, particularly the primary neural cell cultures and neural stem cells. Also, we used to differentiate the neural stem cells into neurons, osteoglia, and so on for the tissue, neural tissue regeneration purposes, and also microglia cells. And on the other hand, so far I discussed about neurons are not responsible. On the other hand, what we used to we functionalized with the RGD cyclic RGD peptide, which is more specific receptor-based, integrin receptor-specific peptide. We functionalized these particular peptides on the polystyrene nanoparticles, and we exposed onto the neurons, primary neurons. As you can see, the neurons, neuronal body was nicely decorated by this the fluorescently labeled polystyrene nanoparticles. So this is a main example of receptor-based, as I mentioned in the mechanism slide. So if you functionalize more specific peptides or functionalize specific aptamers or nucleic acids, you can able to target more specific cell types. And last talk so far we discussed about like nanomedicine, personalized medicine, neurological disorders, and non-invasive, how it is non-invasive and minimally invasive approaches. And my last part, I'm also going to talk about nanosafety, like nanotoxicology. Because so far we discussed about nanomedicine point of view. We should talk about nanotoxicology point of view also, because there are a lot of nanomaterials is toxic. And in our experience, the endotoxins, how probably as a pharmaceutical scientist, you know about lipopolysaccharide is endotoxins, particularly in the parental formulations. It's a problem not only for the parental formulation, it's a problem for even for the nanomedicine research. In our research, what happens when we synthesized our nanoparticles in more sterile environments, even those particles got contaminated by the endotoxins. The polystyrene nanoparticles, two different functionalized carboxylated PEG, as you can see in PEG functionalized, it got huge endotoxin contamination. So why it should be, important, because endotoxins, it's like polysaccharide, it activates the macrophages. When you're introducing into circulation, systemic circulations or brain, it has a lot of macrophages, and the same like coronavirus spike proteins, it contains glycoprotein something. So the same, this endotoxin is nothing but is one of the glycan proteins. That's a problem for this is a problem for our it creates a sepsis, and it is a problem for the human body. And another case about nanosafety. As you know, it's a big question actually whether silver nanoparticles is toxic or not. Many formulations contain silver, because silver is like a good antibacterial, and many people are reporting antiviral and etc. etc. So that's why actually the socks silver, to prevent the bad smell in the socks, they are using a silver nanoparticles. And also mask will be created using a silver nanoparticles, and a lot of bandages function because of the antibacterial natures. And sunscreen, cosmetics, even zinc oxide also in calamine lotions. They're using zinc oxide nanoparticles and silver. A lot of ointments, they're using silver. But the big question arises, when this man, like chemistry behind this blue man, the specific term is called Algeria. This man, this unusual man, what he did, he consumed huge amount of silver ions. At a later stage, what happened is his skin got like bluish, and the specific term is called Algeria because silver ions is bound on hemoglobin, and it creates the bluish color. And also nanoparticles in central nervous tissue. That's another important aspects we need to consider. For example, I'm again emphasizing this the coronavirus situation. So its main entry point is NOS. So even the nanoparticles, for example, these days we are utilizing nanoparticles in many aspects. For example, the urgent need for assessment of biological effects and health risks, acceleration of both manufacturing skills and intention to use. A lot of daily usage we are using in nanomaterials, not only nanomedicine purpose, even for the general energy purpose, many people started using nanomaterials. So imaging, drug delivery. So unintentional exposures may create a problems actually. I'm not blaming all the nanoparticles, but certain nanoparticles like metal nanoparticles, for example, quantum dots, it's creating a lot of problems. Metals, iron oxide nanoparticles, it should be considered actually. And main entry point is like a NOS, and it can enter into brain or in other parts of the body. And as I mentioned, your body is full of not only brain, even other parts of the body also is full of macrophages like immune cells, monocytes. So this is really problematic. So we should consider the nanotoxicology, nanosafety is a very important parameters, and routes of invasion, permeation, and distribution in various cellular and tissue compartments, and effects on different types of neural tissue types that should be explored. And European Union is spending a lot of money on nanosafety and nanotoxicology, and they are exploring in this particular aspects. And other things, why I'm emphasizing the brain. There's a long back, 10 years back, there was a study actually. They performed, they injected nanoparticles, particularly the silver nanoparticles into the brain. After 24 hours, what happened? They found the silver nanoparticles in the brain. But, they found only silver ions, not on particles actually. But what happens, this kind of inorganic nanoparticle, that's why I mentioned inorganic nanoparticles should be considered, because inorganic nanoparticle, when it enters into the body has a lot of different pH, different ionic strengths. So this ionic nanoparticles, it gets ionized, and that's what they found in the research actually. They found a silver ions, but they couldn't find a silver nanoparticles in the brain. So it should be considered, particularly when you're using iron oxide or any metal nanoparticles, so safety. And I think I'm finishing as more early. So I'd like to thank my group in Technion. Recently I shifted to Portugal. So Technion is a Mediterranean Institute of Technology. It's a MIT of Mediterranean. And I'd like to thank the European Commission for funding our research and Marie Curie actions for the prestigious fellowship, and Teva Pharmaceutical. Hopefully everyone knows about this Teva Pharmaceutical. Israeli based, but it located in more than 90 countries in the world. They funded us for the nose to brain transport studies, and all our partners all around Europe. And I'm looking for a widely collaborative, successful work. And thank you all for your kind attention. Thank you so much for this wonderful session and invitation. Thank you so much.
Sir, thank you, sir, for your excellent presentation.
Thank you, ma'am. Thank you so much.
Sir, few questions is raised in the chat box, sir.
Yeah.
Shall I proceed, sir?
Yeah, please, ma'am.
While surface engineering polymeric nanoparticles, one of the common method employed for dogging phagocytosis, is used in PEG. Recently, anti-PEG antibodies have been reported to reduce the therapeutic benefits of PE-glycolated nanoparticles. Do anti-PEG antibodies are really potential?
Sorry, ma'am, I cannot.
1 second, sir.
Yeah.
Do anti-PEG antibodies are really influential?
Okay, so they're asking anti-PEG antibodies are really influential.
Yes, sir.
Actually, one thing I'd say, PEG itself, it's like more biocompatible. Many people are saying actually, but the main problem with PEG, it acts. First I answered the PEG, and then I'll go back to the anti-PEG antibodies. The PEG main problem, why they're using anti-PEG, the PEG main problem is, many reports they are saying that PEG is like a biocompatible, but the problem is, PEG has like a different chain length. So PEG, it acts according to the length actually. So sometimes the smaller PEG smaller molecular weight PEGs are like more biocompatible than larger ones. So I think using a PEG antibody, it can overcome the what's the problems the PEG polymers is facing actually. I'll not assure you like 100%, but it might give some influence on the particular things. Yeah.
So next question, sir.
Yeah.
Titanium nanoparticles, they are really beneficial. What is your opinion on pure metal nanoparticle rather than combined metal nanoparticle?
I think I already answered in my presentation. Mostly, the titanium dioxide, it's really toxic. Many reports are there actually. I think long back you came across one of the Japanese group. They performed research on titanium dioxide nanoparticles, and they found they didn't perform on humans, but they performed on rodents. So they found titanium dioxide nanoparticles are creating offspring toxicity. I mean, they injected the titanium dioxide nanoparticles into the pregnant mice. Offspring, the pups, most of the pups, they bond with like different diseases like acromegaly, like short leg, short hand, some kind of thing. So it's a big controversy when you're using in the metal nanoparticles, particularly titanium and all. When you're using a combined, again, it's a problematic actually. As I mentioned, our body is like has different ionic strength. So when metal enters, metal is not stable inside our body. On the other hand, these days, a lot of research, particularly using titanium dioxide on localized delivery. Localized delivery. Just they are targeting only on localized aspects. Even we also performed one kind of localized delivery aspects. But still, it needs to be proved. But so far, it has been proved that it's a toxic. But still, a lot of research should be done on the particular area of research.
Thank you, sir. Sir, one more question, sir.
Yeah.
Is it possible to estimate the concentration of any neurotransmitter in brain?
What transmitter?
Concentration of any neurotransmitter.
I think it's possible actually using these days is possible because a lot of people are analyzing I mean, they are analyzing the cerebrospinal fluid, and a lot of invasive approaches are there actually. So it is possible to analyze, but in case of humans, it needs a lot of ethical issues. You need to, it needs to be ethically cleared. So it is possible.
The next question. What are the side effects of silver nanoparticle formulations?
That's what I mentioned. a lot of side effects has been reported previously. For example, silver nanoparticles is producing a genotoxicity. It's giving a toxic to the DNA that has been proved in many publications. Of course, many publications, they are talking about application point of view, like antibacterials, even I saw many ointments. But in some cases, it also reported like genotoxicity. Also in my presentation, I mentioned this silver ions, it causes like bluish skin. So these are the toxic. And another problem with nano, when it's nano size, it has like a more surface active, right? So when you're injecting into the body, what happens? Your body is full of macromolecules. In the macromolecules, it gets like accumulated or aggregated. And in many aspects, it's not even, you do not know the biodegradability one aspect, and whether it's the silver, how it is excreting from our body. That's another problematic actually. So I think it's so far it has been proved that it is toxic.
Next question, sir.
Yeah.
Is it possible to formulate nano-vaccine preparation for COVID-19?
There's still a lot of research is going on. Exactly, for example, this Moderna, actually, the famous Moderna, they are developing mRNA vaccine. It's a kind of nano-based vaccines only. It is possible, but we need to consider the pathological aspects of the virus also, because virus is all the time is mutation happening. So we don't know like how you can able to develop the vaccine using a nano vaccines, but we do not know like how this effectivity is in the later stage. So that's a big question always. A lot of people are trying actually on nano-based vaccines.
Thank you, sir. Next question. Is it possible to convert crude herbal extract to nanoform? Still many works are going on and gets published.
Yeah, actually, even my last long back my presentation, the same question has been arised. It is possible. The recently the herbal formulation has like many mix up actually. We can able to convert using a lot of microfluidics technology. A lot of microfluidic technology out there, but the problem is, as I mentioned, it's like a mixture. It's not like a single, like it's not like particularly alkaloids. It's not particularly, it's like a mix of many materials, and you can able to convert using a microfluidic technology. There are a lot of approaches out there. Even we also trying pure drug nanoparticles approaches. You can exactly convert the drug materials into the nanoform. The similar way, you also can able to convert this herbal formulation. The main question is, it has like a mixture, and what is the purpose of converting into nano? You do not know like what is the effect actually.
Okay, sir. So next question. Nano silver is used in exterior paints.
Yeah.
And whether there is possibility for release of nanoparticles from the paints, and if so, are they endangered humans and the environment?
I think again, I answered even presentation also, and I answered for the previous question the same thing. Yes, of course. That's what I presented the nanotoxicology, nanosafety aspects. Like last slide, it definitely dangers the human. Then the last slide I mentioned, nose to brain transport. Every day we are inhaling millions of ultrafine nanoparticles. So when it accumulates inside our body, the problem with nano, as I mentioned, it's a surface active, and it's not excreting from our body. It's aggregating inside our body. It's called a protein corona. It's a biocorona. Macromolecules. It adsorbs on the nanoparticle surfaces inside our body. Even blood itself has a lot of number of more than 5,000, 6,000 proteins actually. So it definitely it is dangerous to our body actually. It aggregates and creates a lot of problems. Silver, not only silver, many metals, many toxic materials.
Sir, next question, sir. So why women are mostly affected by multiple sclerosis, and why it mostly affects the gray matter, why not the white matter?
I don't know the answer for this specific question actually. So I don't know that mechanistic based things. I'm sorry about this. Yeah.
Sir, thank you, sir, for your excellent presentation, accepting our invitation and rendering an excellent presentation on the topic, nose to brain transport of nanomaterials. It was very useful and interesting for us. I thank you once again, sir.
I also thank the management and Hari sir for inviting me. Thank you so much. I hope stay safe in this pandemic.
Thank you, sir.
Thank you, Dr. Murali.
Thank you, sir.
Now I request Dr. Nandakumar, sir. Now I request Dr. Hemlata ma'am to deliver the valedictory note for our faculty development program.
Good evening, dear participants. After 4 days of significant learning process, we have now reached the completion of the online faculty development program on trend and advancements in pharmaceutical sciences. At the outset, I would like to thank our founder, Chancellor, Guru A.C. Shanmugam sir, Honorable President, Dr. A.C.S. Varun Kumar, and Honorable Secretary, Thiru A. Ravikumar sir, who had been most compassionate about the conduction of this program, and thank them for the support and encouragement. I also thank our respected Vice Chancellor for her guidance and for rendering the keynote address during the inaugural session. Prior to this online FDP program, we organized a webinar series of 5 webinars by eminent speakers, for which we received a remarkable response from participants across the nation. With that enthusiasm, we organized this FDP program on trends and advancements in pharmaceutical sciences with significant reciprocation from teachers of pharmacy from various institutions across the nation. Eminent speakers from reputed organizations from India and abroad have given the resourceful talk on most happening topics in the field of pharmaceutical sciences. About 8 technical sessions were conducted over a period of 4 days, and the online feedback and assessment were recorded to ensure the value of the program. I thank all the speakers for their knowledge and experience. I would like to render a special word of thanks to our beloved principal, Dr. N. Harikrishnan, who had been the inspiration and strength behind the conduction of this program. I also thank the organizing committee, Dr. S. Nandakumar and Mrs. S. Gejalakshmi, for their untiring effort and continuous support throughout the planning and execution of the FDP program. We also thank our dear colleagues and students who backed us in the conduction of the FDP. Any program will not be successful without the support of its participants. I thank all the participants who actively participated, learned, and reviewed their knowledge through this program. Hope all the participants had an immense learning during this faculty development program. I thank you one and all. Please wait for a few more minutes. An important announcement regarding the issue of e-certificates. Registered candidates who has a minimum of 50% attendance as well as aggregate assignment are eligible for obtaining the e-certificates. The eligible candidates will receive a mail with link for the certificate form on 15th June. It should be filled and sent before 16th June 5 p.m. The certificates will be released from 16th June, that is on Tuesday, and all eligible candidates will receive the certificate to a registered mail ID before 20th June 2020. So I kind request, please do not mail, call, or send messages regarding e-certificates till 20th June. Also personal messages through to the organizers. If you have filled certificate form and do not receive e-certificate even after 20th June, then you can communicate to the organizers. So until then, stay safe and stay home. Thank you for all your cooperation. Thank you, one and all.